is in the spermatogonia, which is really near the basement area. I'm going then to show you the meiotic process. All of you must have learned this at school. And I'm just going to remind you that this process of meiosis only occurs in the male in the spermatocytes, not in any other cells in the body. So before meiosis takes place, the diploid spermatogonia, the DNA undergoes replication. So each of the chromosome has two sister, you can see two sister chromatids. The blue sort of represents the chromosome that comes from the male, and this one, the pink, comes from the female. It is really important that these two homologous chromosome come together, undergoing pairing and synapses. And then you can see there is genetic recombination so that the resultant cell gets the genetic information from both the father and the mother. And then they separate, and you can see here the chromosome here is different from the original chromosome because now there is reshuffling of the genes, so they contain both the maternal and paternal genes. Then the cell undergoes first meiosis, which is a separation of the homologous chromosome into two separate cells, each with a chromosome, and each chromosome still has the two chromatids. This process of meiosis 1 takes a long time in terms of days. Then they rapidly undergo meiosis 2, which is an equatorial division, so that the resultant cells will be 4 haploid cells, which they will only have, each of one will have a chromatin. And you can see that each of one have been different and it's different from these two chromatin that comes from that spermatogonia. So one cell becomes 4. Instead of having two chromosomes, they have only one chromosome with one chromatid. This process of meiosis only happens in the spermatocyte, in the male germ cells, because you need the haploid cell to fuse with the oocyte, who will also undergo meiosis too, so that it will be haploid. So after so they become after the separation of the chromatids, they become round cells. So round cells are called round spermatids. Then they undergo morphological changes to become to become a mature sperm with a head, midpiece, and tail. This process is called spermiogenesis. There's no further cell division from this onwards. Now, the seminiferous tubules and epithelium of this seminiferous tubule are arranged in this stages of spermatogenesis, and this is a wave, so that the immature, these are the spermatogonia, go to the more mature spermatogonia differentiation, and these are all the spermatocytes. You can see it takes a long time, and then this is the meiosis tool, which takes very quickly to become a round spermatid. So in the rat and in the mouse, there is specific cell association, cell association with each of the stages of the seminiferous tubule epithelium cycle. So if you look at this slide, which is a drawing, you can see that this is one stage where the sperm has these group of cells have a spermatogonia. It has a preleptocene PL, preleptocene spermatocyte. Then it has a pachytene spermatocyte, which is the characteristic big cells with little cytoplasm. And then they go in to become round cells with inside is the sperm, but it has cytoplasm around. And then this is a red sperm. You can see it's a hook-like sperm. And so you can see that this cell association is always the same in one cross-section of the spermatogenesis. So in the red, if you see these cell association, this is stage 8 or stage 7. This is important because these two stages are at the stage of, at stage A is where the sperm is released into lumen, and these two stages are under the hormonal control that Dr. Swertloff talked to you about last time. In the human, it's a little bit more complicated. It's not like a wave that goes from one stage to another. It's spiral. It's spiral. So the seminiferous tubule, if it's like a tube like this, you can see these are tubes. So the sperm, instead of going every cell association, is different at, if you cut the cross section, you have all sorts of stages. In the human, it's sort of classified into 6. So you can see here from the A dark, which is this one, it becomes an A pale spermatogonia still. Then it decided it is going to differentiate, and then it goes into this spermatocyte. This is where meiosis takes place. First meiosis, and then very quickly into the second meiosis to form these round cells, which are the round spermatid. Then spermiogenesis occurs, change in shape, and then at this stage, the sperm is released into the lumen of the seminiferous tubules. In the human, this cycle that is color takes 74 days, and this is a fixed biological constant. It's very difficult to change this as this time. So in addition, the sperm has to go through the epididymis before it's ejaculated. So for anything that affects spermatogenesis, you would take approximately 74 plus 14 days, roughly 90 days, 3 months before it becomes, you can see the effect of the medication or on the sperm. This is important to note with some of you that are going into our group that we are going to talk about male contraception. So I want to show you pictures of what it looks like. This is a human normal testis. You can see here that the spermatogonia are these dark cells that are right next at the basement membrane of the seminiferous tubule. In between here are the Sertoli cells, the nurturing cells, and in between the seminiferous tubules are the Leydig cells that makes the testosterone. Absolutely important that the testosterone needs to be next to the seminiferous tubule, because the testosterone inside the seminiferous tubules, the concentration is very high and is necessary for sperm to develop. And then you can see here are the big cells. These are the spermatocytes. And then the little ones are the round spermatids. And you can see these are the mature spermatozoa. These are two pictures from infertile men. You can see here the seminiferous tubules are atrophic, much smaller. And you can, in here, there's no germ cells. All you see are the supporting cells or the nurturing cells, the Sertoli cell. This man is going to be infertile and nothing can help him to become fertile. This is an, if he's due to a damage to the testis. And this one is also infertile. And in some tubules, you can see atrophic Sertoli cell only, but in others, the sperm maturation is stopped. It's usually stopped at the big cells or the spermatocytes, and this is called maturation arrest. So these are the testis biopsy that is taken from men that are infertile. So question again, post-test.
Okay. Post-test question. How many chromosomes does the sperm have?
Poll quickly. Please submit. We need 100% here.
You heard your teacher. Okay. Results.
97, almost all.
Very good.
Very good. Really, very good.
Good.
Okay. All right. So now we then, I'll show you what it is. Sorry about this. I'm fiddling with my pointer.
Great.
Okay. Another thing. Good. So this slide, we show you what the sperm looks like. It has a head, it has a meat piece, and it has a tail. And each part is important. The head contains the DNA, the genetic material within the nucleus. This is the acrosome cap. It's an acrosome cap that covers the head of the sperm. Then this is the meat piece with mitochondria. Lots and lots of mitochondria coils. And the tail, this is called the tail or the flagellum. The upper picture show you electron microscope of a human sperm that is oval. Human sperm is oval. As I have shown you before, this is a rodent sperm. This happened to be a mouse sperm, and you can see that it has a hook. It has a hook. And this is guinea pig sperm. It doesn't have a hook, but it is flattened. So these are showing you the different, although they have the same function, but in different species, they are different. Okay, I'm having trouble with my mouse. Okay, I need to go. No. Okay, so this is another picture of the sperm, showing the sperm head that's covered by a plasma membrane, and then the acrosome cap here, nucleus with the DNA, the mitochondria that's surrounding the inside of the beginning of the tail, which is called the axoneme. Inside the axoneme, there are 9 tiny arms. Those of you who are going to work with Dr. Wei Yan from our center, the PI of our center, he will talk about the importance of these tiny arms. It's absolutely important the sperm tail has the right composition of the axoneme and the fibrous sheath so it can move around, and the energy for the moving around comes from the mitochondria, and the sperm head is mainly for fertilization and the transfer of genetic material during the period of fertilization. So the sperm then goes through the reproductive system. So they are released into the lumen. It goes through very little tube, tiny tubes called Evans ducts. Then it goes through the epididymis. It's really important that they go through the epididymis because it acquires protein on the sperm surface, and after the sperm passes through the epididymis, they are motile. When they come from the testes, they are not motile, but afterwards, when they come out from the epididymis, they become motile, and the ejaculatory ducts then go to get fluid from the seminal vesicle and the prostate, so that the semen that is ejaculated contains fluid mainly from the seminal vesicles and the prostate, and of course the sperm that comes from the testes. So this is the passage of sperm through the reproductive system. Okay. Can we have a poll?
Okay. Pre-test question. Someone needs to mute someone needs to mute themselves, please. Thank you. Okay, next question. Can a sperm fertilize? Can a sperm fertilize an egg when it leaves the testis during natural conception? Your answers?
Is it just like outside of the testes?
Yes. When it's ejaculated in the seminal fluid, right? We are talking about the sperm in the seminal fluid during natural conception. It should go to the female reproductive tract, right? Okay. So 63% say yes, 37% say no, and we will talk about this process. So the sperm that comes out from the testis, in fact, under natural condition, has to go spend time in the female reproductive tract before it acquire the capability to penetrate the sonar pellucida, which is shown here. This is an egg with a sonar pellucida and the cumulus complex, which are cells. These are the granular cells from the ovary. So the sperm has to go through these, first thing, it has to swim up the female reproductive tract, and then it has to penetrate this vestments of the egg. In order to do that, it has to acquire something called hyperactivated motility. So when the sperm goes down, and if you see a semen analysis, you're welcome to our lab to see it, you'll see that the sperm moving forwards, progressive motility. However, when it goes to near the egg, it has to go through these barriers, and it acquires something called the hyperactivated motility, which means the flagellism, instead of just going like this, goes in this roundabout movement, so that it can go with high energy to penetrate the cumulus oocyte complex and the sonar pellucida, so that it can attach to the cell membrane. So one process is called capacitation. The second process that they must undergo is called the acrosome reaction, the cap. You remember the sperm head with the nucleus and DNA is covered by the cap. The acrosome cap has an outer and inner acrosome membrane, and you can see these are the two membranes then fuse together, release the sperm head, that the sperm head then is ready to get undergo, that can fertilize an egg. So the acrosome cap needs to be removed before the sperm can fertilize an egg. There are many processes that can facilitate acrosome reaction, which is listed here. I'm not going to go through them, but the acrosome reaction is a lysis of the acrosome that occurs in the oviduct in a mouse or a rodent before, and fallopian tube in the females, before spermatozoa can enter the ampulla to allow the sperm egg membrane fusion to take place. If the cap is not removed, the sperm is not capable of fertilization. So this picture it shows in here a mouse oviduct. This is the uterus. The sperm go from the vagina to the cervix, through the cervix, then into the uterus, and then it goes to this uterine tubal junction that is very tight. And then it travels along the fallopian tube, so-called the lower isthmus, upper isthmus, and the ampulla. The ovary is around there, and during ovulation, the egg is released and is in the ampulla waiting to be fertilized. So the investigators here from Japan, very well known, stain the sperm with a green fluorescent protein staining the acrosome cap, and a red fluorescent protein staining the mid piece of the sperm. So if the sperm has an acrosome, you will have a red and green sperm. If the sperm lose this acrosome, it will be red sperm. It then shows a electron microscope of this tight junction that the sperm has to go through. And this is micrograph showing that you can identify these red sperms going up in the fallopian tube. He showed two pictures, E and F, in the lower or at the fallopian tube or the oviduct that's very close to ampulla. And what they want to show here is at this region, which is a little bit further from the ampulla, there are 5 sperm, red, right? Red body. However, every body has a green cap except one. If you move along closer to the egg, there are 3 sperm here, 1, 2, 3. This one has a green cap. Green cap, that means acrosome is not reacted. And these two have no green. You cannot see green. So these are acrosome reacted. And the bottom line is that it's really important that this acrosome, what they show here is that the acrosome reaction takes place before the sperm enters the ampulla to undergo fertilization. I think we have the poll again. So
Yes. Post-test question. Okay. Same question. Can a sperm fertilize an egg when it leaves the testis during natural conception?
100%. Right.
Results.
58%. So the sperm, in fact, cannot fertilize an egg in the natural conception. It has to go through the female reproductive tract, acquire hyperactivated motility and acrosome reaction before it can fertilize the egg. This is totally different. I think some of you may be thinking of fertilization in a tube. In the test tube, you can make a sperm undergo both procedures just by adding calcium or changing the pH. But natural conception, the sperm has to go through the female reproductive tract and acquire those two properties before they can fertilize an egg. Okay, okay. Pull off so that I can go on. Can you take the poll off? I just have 2 more slides to talk about fertilization. Okay, so this is a process of fertilization, relatively simple compared the journey that the sperm has to go through and the multiple changes that the sperm has to go through. The fertilization involves the binding of the sperm to the zona pellucida. This is species specific, means rat to rat, human to human. Then the sperm penetrates the zona hyperactivated motility. This sperm already is acrosome reacted. Zona penetration, and then it touches the sperm membrane. The binding of the sperm to the oocyte, the interaction between the plasma membrane of the sperm and the oocyte is also species specific. And once it binds to the big egg, it fuses. And this, the fusion of the sperm and the egg, is fertilization. And from then it forms a zygote and then an embryo. When the sperm fuses with the ollama or with the oocyte, there are cortical granules that are present in the oocyte that are released. It's released in the perivitaline space, which blocks any other sperm coming in. It blocks polyspermy. My last slide is going to show you just the whole process again. The egg ovulates, the ovary ovulates one single egg, usually at one time during ovulation, and it is deposited in the ampulla with this protective vestments, the zona pellucida, and the cumulus. The sperm comes in, fusion of the egg and sperm forms a zygote. So it's here. Then the zygote undergo multiple cell divisions, moving along the fallopian tube until it reaches the uterus. When it reaches the uterus, it's already a mass of cells called the morula. And then it undergoes a transformation to form, this is where the morula is, or the inner germs mass, and the blastocyst acquires fluid. This blastocyst is where implantation takes place. Once the blastocyst is implanted, it then goes to develop into an embryo. So this is the process of ovulation, fertilization, fusion of the egg, cell division, and finally implantation. I am done. Thank you so much. So questions? I'm going to stop sharing too, so you can, so Ms.Kawen can see you all.
Okay. See us all?
Can you see them?
Yes, I can see them now.
And if we change them, yeah.
You have many questions. Okay. Now, is it Francis?
Yeah, thank you. Thank you
The
so
floor
much.
is yours. I'm sorry. No, go ahead. The floor is yours.
Oh, thank you. Thank you so much for your lecture. It was really interesting. But so I remember you said that in order for the sperm to actually be able to fertilize the egg, it has to go through or it has to spend time in the female reproductive system. I was wondering why that is. Like, what makes it go into that stage of the what's it called?
Okay, so these processes, the sperm then has a long way to go, right? It first has to go through the vagina, the cervix, then into the uterus, and then you go, during each of the process, you know that millions of sperm, about let's say 60 or 80 millions of sperm are ejaculated into the female reproductive tract with each sexual intercourse. So in humans. So during the process, when it goes up the female reproductive tract, some of the sperm are eliminated. And then when it goes to the the fallopian tube or the oviduct, it requires it already changes the way that it moves when it's really close to the egg. At the same time, it undergoes the acrosome reaction. These are the processes to make sure the sperm can fuse the egg. As it travels along the oviduct, again, more and more sperm are weeded out, so that at the end, you only want one sperm to fertilize the egg. So the fertilization of the egg, if you one sperm fertilize the egg, it prevents further sperm going in because you don't want polyspermy. That's abnormal fertilization. So this is the process that that the sperm has to undergo before it can fertilize an egg. In vitro, in vitro fertilization, when the sperm is taken out from the seminiferous tubule, from the semen or from the seminiferous tubule, then the sperm is put into an artificial medium. In the artificial medium, capacitation and hyperactivated motility can be induced. However, hyperactivated motility usually is not necessary because the sperm is placed directly next to the egg or injected into the egg. So that process is not necessary. However, the acrosome reaction has to take place. Okay.
So what makes a sperm go into hyperactive motility?
Yes, usually there are many different factors, but the commonest thing is calcium. The commonest stimulus is calcium. It's also when the pH changes from an acidic to alkaline pH, it also makes it undergo hyperactivated motility. And when it's really close to the egg or close in the ampler region, progesterone of the female hormone progesterone also can induce hyperactivated motility. So there are multiple factors to make sure that this goes place when the egg is up, when the sperm is going through a lot, right? It has to travel a long time to make sure that sperm is ready to fertilize.
Okay. Thank you so much.
Thank you for your question. Next, I hope I pronounced Neveneth. Neveneth.
Yeah, it's Navneet. I had a similar question for the acrosome reaction that you mentioned. What is like, is there like one like dominant factor that causes the acrosome reaction to start, like calcium um for the other one?
Okay. So many of this, you can see the list of substances is very long, right? It can be initially they thought it's the binding to the zona pellucida or in the cumulus cells that induces the acrosome reaction. However, the more recent studies that I show you, the red and green sperm, it shows that in fact the zona pellucida and the cells around it is not the main factor. So which factor actually happens? does not know. It can be tyrosine phosphorylation, it can be calcium, it can be changes in the pH, it can be proteins that are surrounded by the surrounding the sperm. We except for each sperm we don't know. All we know is that you do not need the egg to get the acrosome reaction, and that was only proven in the 2000 in around 2013, when it was shown that the sperm doesn't need to penetrate the sonar to get acrosome reacted. So this is changed. So the exact thing, I don't think we know. So you can see there are numerous factors there. Those are experiments in vitro that you can put something in and the acrosome disappears, right? Okay.
Thank you for your question. Next is Vivian. Please ask your question.
Yes, thank you so much for your talk. I had one question. So I read that in the oviduct, the isthmus holds on, like gradually releases the sperm to prevent polyspermy. So what do the biological mechanisms look like? How does it gradually release the sperm?
I think what you're talking about is that you don't want to have too many sperm going forwards. From the same group in Japan, what they have seen, what they have shown in the they have a microscope that you can look through the wall, the very thin wall of the oviduct. They can see, in fact, less and less sperm go up, so that they do not know the mechanism, but one sperm will go up and the rest of the sperm is held back. The exact reason is not really known, as far as I know.
I see. Thank you
Very
so
good.
much.
Good
Hi,
point.
Grace. Your question, please.
Yeah, so thank you so much for your talk. I really enjoyed it. I read that there's like Klinefelter syndrome, so where like males have XXY instead of XY, and that it sometimes causes I guess like infertility issues. So I was wondering like what's the process behind that, and like does it lead to the same, I guess, spermatogenesis arrest that we talked about?
Yes, really good question. Dr. Swerlov show you, and in the Klinefelter syndrome patients, let's say most of them will be like the picture I show you on the right top, where you have seminiferous tubules are empty and atrophic, and as the man grows older, all you see is highlighted tubules with no germ cells. However, in the actual circumstances, about maybe 25, maybe up to 50% of the men, they don't have a single sperm in the ejaculate, in the semen. However, when the urologists go and look at the testes under the microscope, they can sometimes see a pocket of sperm. The tubules become slightly bigger, and if they go in, they get one sperm. So if they get that one sperm, they can fertilize an egg. So Klinefelter syndrome, 98 most of them are isospermic, and they were supposedly to be sterile, infertile. Now with the assisted reproductive techniques, with the female undergoing stimulation so that you have the egg ready in a test tube, then the surgeons go in and get the single sperm. The fertilization, provided they get the single sperm and inject it into the egg, they are injected directly into egg, then the fertilization rate is almost like any type of in vitro fertilization. So that the take-home baby rate, if the man, if they can find a few sperm in the testes, the take-home baby rate is almost like 25%. So with this assisted reproductive technology, they converted a totally infertile, sterile man that we used to see in the past to become, some of them can become fertile. So that obviously this is the advance of assisted reproductive technology. We learn a lot. However, it's also a barrier for reproductive biologists, and because the couples would not like you to do anything, and they are not, people are not so interested in really understanding what's going on. So in XX, everybody believe is the extra X. The extra X in the maybe the reason why that the sperm would not mature, because in the testes of the Klinefelter man that have that you can see pockets of sperm, it is believed that those sperms are haploid and it does not have an XX or XY. Okay, so that they are they think those one or two sperm in fact is normal haploid, not diploid. So exact mechanism of how the X does that we don't know. And clearly the X doesn't undergo X inactivation, it becomes like a female, and it controls a large number of genes, and they don't know which one is happening. So these are some of the things like Klinefelter syndrome. We should have done more studies, but the man obviously wanted to have a kid, so they are not going to let you do studies on it in our here, and also in probably in Germany. We have a mouse model that is XXY, but the generation of that mouse model is very, very complicated. My colleague here, Dr. Lu, can generate this, and we use that mouse to study the mechanism, but it's not so easy, difficult to generate.
Thank you.
Thank you. Okay, Jed. Hi, Jed. Your
Hi.
turn.
Thank you so much for your presentation. It was really awesome to learn more about male reproductive system and all that. So I was wondering, I know some couples have like problems conceiving children partly because of some men having like poor sperm motility. So is that condition more genetic or environmental or both, or is it cause like a problem in like spermatogenesis or in some of the structures of like the male reproductive?
Okay, in the so some of them are due to the hormonal causes that Dr. Swertloff talked about. Those things we can fix. Most of them we can fix, not Klinefelter syndrome, but we can fix. The other, in the past, they have been called, we don't know the reason why, but the recent study, and more recent study, and maybe you'll hear from Dr. Wei Yan, that more and more studies show that this is a genetic defect. Does environment place? It probably does. So that the common, but the, so if you are, obesity is one of the things that we are studying, and we're studying epigenetic changes. But obesity, does it decrease fertilization? Some studies say yes, some studies say no. Some say that bariatric surgery works, some doesn't. So it is not very clear. However, the belief is that if the spermatogenesis is already defective for one reason or another, and you add on the environmental factors, you're exposed to the phthalates, you are exposed to heat, the man is obese, you are smoking, then you decreases the chance of fertility. So is nurture and nature. Nurture is epigenetics, environmental is genetics. So it's a combination of both. This is a problem for us unless there's a breakthrough, unless there's a big breakthrough. Many of the men with the genetic diseases or undiagnosed genetic diseases, there's no treatment. Okay, the treatment is to treat the wife, and you try to find a normal sperm and inject it into the egg of the female partner. That's not treatment of male infertility. So it's a big thing that needs to be studied.
Thank you.
Thank you. Matthew. Hi, Matthew. You're next.
Hi, Dr. Wang. So I was curious. I know you mentioned the words Sertoli cells, spermatogonium cells, spermatocyte, and spermatid. Can you clarify again which stages those are all in?
Okay, so you start from the primordial germ cells, spermatogonia, stem cells. Then they become they are the spermatogonia. You can't distinguish them. The spermatogonia, it depends on whether it's a rat or human. In the human, it is the most primitive cell is called dark spermatozoa, spermatogonia. It becomes pale spermatogonia. Then it becomes a committed spermatogonia. Committed means they are committed to differentiation. That is not a reversible. You can't go back. Once you go to the spermatogonia B, which is committed, then it goes to spermatocyte. There are 5 stages of spermatocyte. The big cells that you see most frequently is the primary spermatocyte. And the biggest one is called the Packeting spermatocyte. So the reason why there are so many spermatocytes is that it has to do the 2 meiosis division. The first takes a long time because you have the chromosome pairing, exchange of material, segregation, division of cells. The second one quickly takes place. And then you end up with a round spermatid. The round spermatid have a little bit of cytoplasm. Then it undergoes morphological changes in the shape to acquire a tail, a midpiece, and then the cytoplasm is lost. So there's essentially no cytoplasm around the nucleus of the sperm. It has an acrosome cap, but it has no cytoplasm. All the energy comes from the mitochondria. It makes ATP to make the sperm move. However, the sperm tail structure has to be intact. And that I think Dr. William may talk about it next time. Okay.
Dr. Wang,
Yes.
can you clarify what the Sertoli cells are?
Okay, the Sertoli cells are cells within the seminiferous tubule that you can't see them in the thing, but they are very obvious if you see a real section. And the Sertoli cells are the cells that are absolutely important for spermatogenesis. They produce, in the past, they call nurturing cells. They produce the nutrients, but they secrete so many different chemicals that are important for spermatogenesis. The Sertoli cells is also where the regulation of spermatogenesis comes from. The regulation of spermatogenesis, Dr. Swertloff told you, depend on 2 things. One is the hormone coming from the pituitary called follicle stimulating hormone. That works on the Sertoli cell. The second, you have the hormones coming from the pituitary called the Leydig cell, but called luteinizing hormone. The luteinizing hormone acts on the Leydig cell, which I show you next to the seminiferous tubule. It produces the male hormone. The male hormone diffuses into the seminiferous tubule, acting on the Sertoli cells. So the Sertoli cells is the regulator of spermatogenesis. No Sertoli cells, no sperm.
Okay. Thanks
Okay,
so much.
if there's no sperm, the Sertoli cell also doesn't function well. So it's interacting cells. The interaction between the cells are important inside the testes.
Got it. Thank you.
Okay.
Thank you. Next we have up, and excuse me if I pronounce your name incorrectly, but Pira? Pira?
Yeah.
Priya. Okay.
So, Dr. Wang, I know you showed a diagram showing spermatogenesis in the rat with the different stages, and I believe you said that stages 7 and 8 were the most important. So, can you reiterate why that is?
Oh, no, all of them are important. You need to go through the whole thing. It is important. I think Matthew asked the question before, because at stage 7 and 8 are very, very sensitive. They are very, very active stage of metabolism. So if you have any stress coming into the testes, you may have ischemia, no blood flow, okay? You may have heat, or you do a chemotherapeutic agent. They're going to hit those cells that are highly active. Moreover, those 2 stages, 7 and 8, are under the control of the hormones that are absolutely important for spermatogenesis, the male hormone and the follicle stimulating hormone. So those stages are easily recognizable, that people can easily study. It's not that the rest are not important, they are still important, but these stages usually are most susceptible to injury. Okay. And also in stage 8 is when the sperm is released. If there's a problem with stage 8 and the sperm is not released, the man is going to be infertile. So in a way, that's why I think that is where the regulation of the spermatogenic cycle occurs in the rat.
Okay, thank you. That makes sense.
Thank you. Next we have your question, please. And if I pronounce your name incorrectly, I apologize. Tayam?
Yeah, hello. Thank you for the lecture. Sorry, my camera isn't working right now, but so I think you talked about a little bit on one of the previous questions, but what I'm curious about is how like about finding healthy sperm. Like more specifically, I'm curious about finding healthy sperm, like the specific process to that in finding HIV tested semen, and like how it works and why it works.
Okay, there has been questions about this, and you're talking about the virus. The HIV virus is not on the sperm. There have been studies suggesting it may be on the sperm. HIV virus is in the white cells in the sperm. So I'm making things simple. So as you go, the sperm released from the testes, it acquires, it has very little fluid, but then it goes through the prostate, it goes through the seminal vesicle, and doing that you acquire fluid. And if there is infection of the genital urinary tract, it acquires the white cell. It's the white cells, predominantly lymphocytes, that carry the HIV. It is not really on the sperm itself. I do not know whether I answer your question. So
No, that answered it. Yeah.
Yes, the white cells are infected. It's not the sperm. Okay.
Okay, thank you. Next, hi, Devin. Please ask your question.
Yes, thank you. I had two quick questions. The first question, it was back to men with Klinefelter syndrome. Do they have bar bodies?
Yes, they have like females, 2 bar bodies.
Oh,
okay. There are 2 bar bodies, and it's something that we usually don't do, because we went straight away to do a chromosomal analysis, because the Klinefelter syndrome, most of them have clinical features that we can either see them clinically, or we run a lab test and we can know that is. So we will run a definitive test instead of doing the bar body. Absolutely right. Like female, 2 bar bodies.
Thank you. And my next quick question was, how do you induce the acrosome reaction when you're doing in vitro fertilization?
Oh,
How
really simple. You put in the sperm there, and then all you have to do is either increase the pH, or you use a calcium ionophore to stimulate calcium release, and the acrosome reaction is gone. Okay, it's very simple.
Well, that sounds easy.
Yeah. It's simple. Or you use.
Thank you.
You induce tyrosine phosphorylation. It does that. You put in a little. Sometimes you put in progesterone in it. You can induce it. Okay. Calvin, I think we have. Are there questions in the chat box?
Thank you so much.
We have questions in the chat box. Okay.
No. We are done, I think. No questions.
Yes, and hold on. Okay. Yes.
Done, right?
Well, we have like 5 minutes left. If anyone has any other questions, please feel free to ask. The floor is yours. 5 minutes, and I will go ding when the 5 minutes is up. Okay, we have Bianca. This is your time, Bianca.
I have a question related to the reading. It said that they found out about all these substances through gene knockout. So is that using CRISPR or other like gene editing technology to figure out like which substances are like necessary for fertilization or spermatogenesis?
Yes, so there's using modern genetic methods. the understanding of the many genes. There are many genes that control spermatogenesis. So using the gene editing by taking out certain genes, then you can see that so you need to first thing you can identify the genes that may be related to infertility. Then you try to knock down the genes, make sure you have the phenotype, and then you try to validate this and find this in human infertility. Right, after all, you want to find the gene so you can understand what is happening and can you treat people, apply it to infertility, or for our instance to to induce infertility for male contraception. So it is a tremendous progress. Is using modern molecular biology techniques applied to both animals and then later on validated in infertile men. And if you find a gene and you validate in the human and you find a person that has that, then it proves that this is really important. So these techniques, I hope Dr. Yang is going to talk about gene editing in his talk next week. He's going to talk about genetics. So I think you can ask more about it. But it has contributed dramatically to the understanding of which gene affects which process. However, they are with these new techniques. Previously, there are only a few, a handful, 11, let's say. The next year becomes 40, and now it's like 80 or 100. So more and more genes are being discovered. That one gene may be causing infertility in one patient, but it may be another. So it's a long way. We have a long way to go still. Okay. Ask Dr. Wei too, please.
Okay, thank you. Next, and I apologize again if I pronounce your name incorrectly. Is it Jenny?
Yeah. I was curious, is it possible to create male contraceptions by like inhibiting the removal of the acrosome?
Yes, yes. So all those are so yes. So all those are possible. Any step that we talk about can be inhibited, and that's what people are looking at. Because if you inhibit something, really good question. Because it inhibits something that's only happening in the sperm, you are not going to cause. Hopefully, you're not going to cause problem elsewhere, right? So this is the target of the National Institute of Health in the drug discovery or the contraceptive discovery branch. That's exactly what they want. They want to have processes that are controlled, that are specific to the testis or to the sperm, that will be targets only on the sperm. We work on hormonal contraception because we know what it is. Hormones work throughout the body, therefore it's not an ideal, but it's a prelude to these specific molecules that inhibit specific processes or proteins or enzymes that's specific to the testis and maturation of the sperm or to the sperm itself. And that's really a good question. That's it. That's our hope for the future. Okay.
Well, I see no more questions, so everyone has all their answers, or you have 1 minute for one more question.
Yeah, good.
Okay. Well, I just like to thank you for all your wonderful questions, as well as you are awesome students. Awesome, awesome.
Very
Well,
good. Yeah.
I applaud you, you know. So, and thank you for your participation. Uh, Dr. Wang, would you
Answer if
anything
you
else?
have, remember, we want feedbacks, we want to improve our series. You get questions from education, you also get questions from our center specifically. So please answer the questions. They will be sent to the questions from our center just started, and it will be sent to you after 5 p.m. tonight. Over the weekend, click on it and answer. There are only 3 questions. We want you to give us feedback so we can improve. Okay?
And go and enjoy your weekend.
Yeah. Thank you, Calvin. Thank you. Thanks, everybody.
Okay, thank you.
Thank you.
Bye.
Thank you so much.
Thank you.
So much. Thank you. Have a good
Good
day.
weekend, everyone.
Yeah, thank you.